I really liked this session. There was such a variety of presentations and a lot to learn. And I had all kinds of questions along the way. I had a few that came to mind. And I guess I had several kind of questions just kind of around the notions of kind of availability and sharing. But let me just get to a few questions that popped up in my mind first, and anyone is welcome to join in. And I'll maybe go sequentially. I had some notes kind of in order. Yeah, I was really intrigued with Dr. Kruger, your presentation. And it was amazing to see the visual analytic capabilities and the ability to interact at different levels. And the lens, I always find the last part, the lens ability, just mind blowing. I was going to ask you a little bit about and this is maybe because I'm a person who works in computation. I work kind of more on the data matrix side. So you showed a few of the data matrices that are the cell by feature matrices, and the interactivity between those was available. I was wondering if you are also exploring capabilities where people would be working with the data matrices in a notebook, say in Python or R, and would be able to interact with in the same kind of way by doing their analysis in R or Python and sending it over to the tool, for example, or receiving data from the lens view. Is that something that you've started thinking about as well, adding to the already cool things that are there?
Yeah, thank you. I think that's a great question. I think like currently the tools that we develop focus more on, let's say, users that are not so much computationally through the visual interface. But I think this is like really a field I think that should be addressed. Like computational biologists who are familiar with code, who usually use the console, but they also need a visual interface at the same time. So really having that somehow integrated is, I think, very important. But we haven't specifically tackled that in one of the tools I presented today. But I think that is really, really important for the future to step forward to do this.
Yeah, thanks. So definitely that would be on a wish list for many people, I think, to have that kind of ability to connect those two things.
Maybe I can add to that the backend of our tool is usually web-based tools, right? So the backend is all in Python, so it shouldn't be too complicated to integrate that, so that you can, like, instead of, trigger the change of a selection or something like that through the interface, you could just send you could just send a code to the backend, so that you could execute that and then see the visual result instead.
Yeah, cool. That's exciting to hear that might be a possibility in the future. I had actually another kind of tools and availability question for Dr. Beacham. I loved your presentation as well. It was really cool to see the capabilities you presented, and I remember working with NanoString data in the past, and I know you had also made available I think it was like R packages or methodologies that interact with that people can install to interact with the data. Is that something? And that was a few years ago, I think, with like nCounter or the earlier technologies. Is that something that's also happening with the spatial profiling, that there are kind of open software as part of the ways to interact with the data?
It's absolutely critical, right? There's so much activity happening. No one's can write all the software we need, right? So on our atomics, what we get is it can import eventually it'll be both R and Python can come in, and then it can be utilized in the same cloud network as all of our other tools, right now only the R side is implemented completely. So you can come up with some custom way that nobody's ever done. We'll call it the Kruger special or something like that. You write it in R, then our Atomics platform works as what's called a pipeline orchestrator. You just connect different elements together and it will run the pipeline. But you could import, something that you wrote, or it could be an ADA special, and then insert it into the pipeline of the manipulations you want to do, and you're doing it at scale. So if it's written in R and somebody stuffed 5 million cells into your algorithm, then let's see whether it can chew up 5 million and give us an answer back. But in terms of the mechanics of everything, it's established. I think it's really important. We got to enable open source on everything. One of the things that NanoString said is that there'll never be an equation in anything I do that's not part of the public domain. So everything we do from a scientific perspective, all of our manipulations are put out into the public domain. So if you don't like the way I divide by 2, you can make it divide by 3 or something. I think that those two things are pretty important.
That's great to hear, and that's great that that's an overall kind of part of the thinking around making those technologies available, and that's super important. I'm going to jump a little bit over to the last presentation, which was also very cool. I thought like the kinds of ways that you had different ways to assess the both like your metrics, but as well as the applying different methods and seeing how, say, differential expression was affected by different things, including making the calls in the wrong direction, something became positive or negative, which was previously negative. And do you, again, in terms of like the availability of software, I imagine that's part of something, and like that you're making methods available as well. And I was going to ask specifically around like the estimates for the cell volumes. Is that something that, say, if it's like important to scale by cell volume, is there like giving that kind of slicing effect that you talked about, is there something in the methods that you make available that would have like a cell volume, estimated kind of cell volume and cell volume correction kind of, is that part of the package as well?
Yeah, that's a really good question. So we, in this particular study, we use only the publicly available data that is available with the spatial data that we're assessing. And different technologies will make different things available. So we actually weren't able to assess cell volume normalization in all of the different data sets we use. Sometimes they only provide cell area, especially if they're segmenting on kind of the maximum projection of the images. And so this is kind of why normalizing by cell volume can be so difficult, either A, because the data is not available, or it's available in a way that makes it hard to segment, or hard to segment on all of the different Z planes that have been imaged in a way that allows you to reconstruct the 3D volume of the cell. And yeah, so maybe kind of now, given that we know the importance of scaling the detected counts by cell volume, then that will be something that is more intentionally provided as more data becomes public.
But I do think that what you brought up was these smaller panels were basically cell typing panels. So it's really a bias. You don't want to be normalizing off of your little tiny panel. That's really, I think that was pretty clear, Lila, in your examples, is that, yeah, you don't try to normalize off a little thing that's less than 500 plex, because it's mainly cell typing is what those genes are doing there.
Yeah, that was kind of fascinating how when you chose like a random set of genes that in some ways it did better for some applications than the targeted, which kind of makes sense in terms of what you were saying, in terms of those being chosen for a particular reason. Yeah, that was kind of some of just randomly kind of writing things down as we heard the presentations, and I was also going to ask Lila, would you, based on the different different normalization strategies? Is there, what are you willing to say or go out there and say that, you know, one of them is like start with one of them in particular? I saw, depending on the figures that you showed, some of them did better in some situations and some of them did better in others. So.
Yeah, I mean, sort of as I presented in the kind of decision tree at the end, it really depends on what data you have. I mean, if you're designing a panel a priori and you want to run analyses on all of the cells in your tissue, then I would probably recommend designing a well-representative panel of the gene expression that you expect in your tissue. If you're working with publicly available data and can't make that decision, then if the cell volume information is available and you're kind of confident in the way it was segmented and confident in the cell volume estimates, then I would recommend using that. And this is also based on things that I think folks in the FISH field have known for a while, that not only is kind of the proportion of cell volume captured not always 100%, but also cell volume is kind of causally related to the gene expression profile of your cells. So there have been studies that have shown that if you artificially increase the cell size of a particular cell, the gene expression in that cell will go up, and so you kind of need to normalize for that. And yeah, and so that is kind of the best case scenario. If you have kind of good cell volume estimates, I would recommend normalizing by that. If you don't, then kind of the subsequent leaves of the decision tree are figuring out if your gene panel is more or less representative, and we are kind of working on metrics on how to quantitatively assess that. And if it is representative, then if your gene panel is big enough, then you can kind of assume that the total genes detected in a cell are basically a good enough surrogate for cell volume. But if they're not, then probably one of the, probably I recommend restricting your analysis to those cells that are well profiled by your gene panel.
Great, thank you all so much. I was noticing we started late, and apologies for we're a little bit over, so we'll try to wrap up this Q&A session. I'd like to thank you all for joining this session. It was really fascinating to see all the things, and Dr. Beacham, thank you for running between buildings here in Seattle.
No problem. We would have fed back that. Oh, we had a feedback loop that would have been terrible if I'd have done it from inside that meeting room. So
Yeah,
really enjoyed all the talk so far this morning. Looking forward to
great. Yeah, thank you.